All right, welcome everybody. Welcome to the session on CFTR modulators and their ability to reduce airway infection and inflammation. I'm Ben Kopp from Emory University. I'm joined by Robert Gray from University of Edinburgh. And we have a really exciting session today for you. Just want to talk about a couple logistics. They have changed the format for us this year, so there will probably be some kinks in this, so please bear with us. And I think a lot of people's slides on the NACFC templates have some changes in titles and weird things that are showing up, so there may be some idiosyncrasies in how the slides appear today. How the session was set up for us is there going to be a series of talks, and at the end we'll have a panel discussion and question and answer period. So we will also, though, just kind of at our own discretion, we may allow one or two questions at the end of each talk. So on your app, as you noticed this morning, there are no microphones. So on your app, if you go to this session, there is a discussion tab and a question and answer tab. And so you can put in your questions throughout the talks there, and then everybody can vote on those. So we'll have a popularity contest today and see whose questions are the best. And we can, we'll help read those out to everybody. But like I said, we may do a question or two at the end of each talk as well, but then we're supposed to have a group think at the end where we can have some discussion. As you can see, they left us a nice set of chairs for our panel discussion up here, so we're going to improvise at the end as well. So we're being flexible today. All right. So as you can see, even though I corrected my slide, it didn't mess up the title, so we have no relevant disclosures. I did want to point out some other relevant programming for those that are coming to this session that relate to CFTR modulators and infection and airway disease. As you notice, some sessions today on microbiome, tomorrow's session on CFTR and innate airway, and as well as a rapid fire poster talk. So those are a couple relevant ones to the talks today to build upon what we're talking about. So we're excited to have 4 talks today, and really kind of focusing on the changing lung environment and the CFTR modulator airway. So we're going to hear from Pradeep about how infections and the bacteria and pathogens in our airway are changing post-Trikafta. We're going to hear from Rob Quinn about the microbial communities and the microbiome and the metabolites they can produce and how this can change in the face of modulators. Then we're going to hear from Mimi Saavedra and really understand the importance of the adaptive immune system and how T cells are affected by metabolites in the airway and how this is being reconstituted. And then finally, Robert's going to end with, how do we to resolve infection and repair our damaged airways in this new era that we're coming to. So we think all these talks will tie nicely together and we'll have a great discussion. So with that, I'm going to welcome up our first speaker, if he is here. I know Pradeep was really busy this morning. He just spoke at Physician Grand Rounds in the last talk, so he's revved up. So Pradeep is a professor at the University of Washington and is going to talk about infections after corrections. We ain't done yet. Welcome.
Thanks. Thanks the organizers very much for the invitation to speak. Start by saying I don't have any disclosures to make. I'm going to talk about, how do you get it going here? Okay. I'm going to structure my talk in 5 questions about CFTR. In fact, not just CFTR modulators, but all my data is going to be reporting about ETI's effects on lung infection and inflammation. And so the first question is, and I'm going to be talking about 2 studies. One is the CFF-sponsored PROMISE study, which is a multi-center trial of real-world ETI use in many centers across the U.S. And the second is a smaller bronchoscopy study that we did in collaboration with the University of Iowa. So the first question is, how does ETI affect sputum microbiology? So I'm going to show you some data on this. So this is from the PROMISE study. And what we found in PROMISE was that there's a really dramatic reduction in the common pathogens that are present in CF sputum. So I'm going to show you first the data for Staph aureus. On the y-axis is plotted CF use per gram by culture of Staph aureus, and the x-axis is plotted time in months. And as you can see, right after starting ETI, within a month, that's the first time point we examined, there's a 100-fold reduction or so of Staph aureus density in the sputum, and then it plateaus out to 24 months. See a very similar response, almost identical in terms of the averages. Response in Pseudomonas aeruginosa, again, y-axis is CFUs, x-axis is time, and these are really remarkable reductions. The mean decrease in Staph aureus at a month was was over 100-fold, 2.4 logs, and very similar for Pseudomonas aeruginosa. See if a similar response when we look at the less common pathogens. So here's the numbers of subjects are much less, but this is Stenotrophomonas multifilia, marked reduction in a month, it remains stable. And this is the data on Aspergillus fumigatus. This is a DNA-based assay that uses digital drop PCR to measure the number of ITS copies present in the sputum. There's between 20 and 50 ITS copies per cell, so it's a very sensitive assay. But nevertheless, you can see that in most cases, by 24 months, Aspergillus fumigatus DNA becomes undetectable. But while these traditional pathogens show marked reductions, other bacteria found in the sputum do not. And so we often think, or in the past thought about the traditional pathogens in CF being these organisms, Pseudomonas, Staph, Steno, Haemophilus, and a few others. Recent microbiome work has shown that there is a large number of other organisms that can be detected in the sputum using DNA-based methods. And so I'm going to show you one piece of data that relates to this, in which we grouped up all the CF pathogens together, these and the others that I mentioned, and all the other bugs that did not qualify or were not categorized as a CF pathogen. And so here's the pathogen data. First, you see about about a 10 or so fold reduction in a month, again, that remains stable. But when we look at the other organisms that are present in the sputum, they're rock stable. And this is true when they're pooled together or if they're examined individually. So individual grafts for Streptococcus species, Veillonella, Rothia, Prevotella, all look the same, no change with ETI. Now we can look at the composition using Bray-Curtis distances of the overall sputum microbial composition. And so what's done here is each sample is compared to every other sample using the Bray-Curtis statistic, and then the data is visualized in this multidimensional scaling method. The gray circles are the pre-ETI data, and then it gets blue, red, or yellow, and green after 6 months of ETI. And what you can see, if you take all the data together, all the sputum taxa that are found by DNA-based analysis, you see that there are these two wings that represent the pre-ETI sample, and then after ETI, the microbial composition converges towards near this origin. Now, if you computationally remove the DNA sequencing reads from CF pathogens. So now I've just taken away all the data from the CF pathogens and repeated this analysis. What I see is no effect. Okay, you got a scattergram before and a scattergram after. And that basically reports the same data I just showed you in a different way, that what's changing in the CF sputum after ETI is the traditional CF pathogens, the other organisms are not. So question 2 is, why do these traditional pathogens decline, but the anaerobes and other oral bacteria don't decline? Well, one possibility is that ETI treats lung defenses that either only or primarily act on CF pathogens and leave these other organisms alone. I think this is probably unlikely. We know that one of the major effects of ETI is to improve mucociliary clearance. This should non-discriminately remove organisms that are present in the lung. We also know that one of the prominent effects of ETI is reduced mucus plugging in the airway. That's been consistently seen on multiple highly effective modulators, a decrease in mucus plugging. That should actually perhaps result in a bigger decrease in the anaerobic bacteria, since the mucus plugged areas are the proposed growth niche for anaerobes in the lung. And here's an example of that. Here's A patient studied by these colleagues who did CT scans before and after modulators, you see a big decline in the mucus plugging, which you would expect would result in decreased anaerobic growth. So I think that's unlikely. Another possibility is that the pathogens are coming from the lungs predominantly. That's where ETI works, whereas the oral bugs and the anaerobes that are commonly found in CF sputum are predominantly coming from the mouth, where ETI may not have any effect. So then you could get this effect where pathogens that are coming from the lungs are reduced by ETI, the oral anaerobes coming from the mouth contributed to the sputum by contamination are not reduced by ETI, and the net effect would be reduced pathogens, but other organisms showing no changes in absolute abundance. Now, if you look by relative abundance, you would certainly, and we have done this, you'll certainly see a change where the pathogens go down and the oral organisms and anaerobes go up, but that's a head fake, I think. It's all a consequence of that metric having to equal 100%. If you look by absolute abundance, as I showed you, the pathogens go down, other organisms don't. So question 3, do we have any evidence that ETI causes eradication of pathogen infections? And unfortunately, the answer to this is it looks like, so far, very few subjects who take ETI become durably pathogen negative after ETI. This is a work in progress because, first of all, just determining whether infection has been truly eradicated is a hard task, especially if you're using non-invasive approaches. But you might get more data about this when we look longer, and I'm only going to show you data out to the first 6 months of ETI use in the PROMISE study. So I'll show you the data first for Pseudomonas. We had 49 subjects who came into the study, Pseudomonas aeruginosa positive by culture, and they provided at least 2 post-ETI samples. So we limited our analysis to those people. There were some disruptions due to the pandemic, but we wanted to have at least 2 negatives. And so if you look at those 49, only 14 became repeatedly culture negative, that all of the samples became negative after the initiating ETI, and they provided at least 2. Now, if we look at those same subjects, those same samples, in fact, using a very sensitive Pseudomonas droplet digital PCR assay, the number decreases further. Only 5 of the 49 were both culture negative repeatedly and PCR negative for Pseudomonas in their sputum. The numbers are a bit larger for Staph aureus. There were 83 subjects who came in positive for Staph aureus and provided at least 2 post-ETI samples. Only 6 of these subjects became repeatedly culture negative for Staph aureus, and only 3 of these subjects were both culture negative and PCR negative in the samples after starting ETI. So question 4, why do most subjects remain infected with their pre-existing pathogen despite really remarkable improvements in CFTR activity induced by ETI? So we were interested in exploring this, and now I'm going to transition to the bronchoscopy study I showed you, and that was performed mainly by Samantha Durfee, and I'll show you a picture at the end. She's a PhD student in my lab. And she was interested in the hypothesis that patients remain infected because infection predominantly persists in the lung regions of each subject that have the most severe structural damage. This would be akin to the situation we think in non-CF bronchiectasis, where you have an area of structural damage, and that's the cause of persistent infection. So the idea here is you have a lot of lung heterogeneity, areas with severe damage, and areas with mild damage, and after ETI, perhaps these mild areas clear, but infection persists only in the severe, and if you're looking at sputum, you'll just see continued positivity, perhaps with lower counts. So to test this idea, Sam devised a study where we sampled the highest and the lowest damaged lung regions within subjects before and after they started ETI. So here's the idea. We had patients undergo CT scans, asked a radiologist to pick the 2 worst regions in that patient's lung, the 3 best regions or lowest damaged regions in each patient's lung. Then we did bronchoscopy of all 5 regions. And we did that before ETI, and we went back to those very same regions 1.5 years after the subjects had taken ETI. Note this is a pretty small study. We were only able to enroll 9 subjects because of the pandemic, but it was still quite informative. And what we found was that ETI produces identical clearance rates and identical changes in PA density in both the very highest and the very lowest disease regions of each subject's lung. So what I'm showing you here on the y-axis is the percent of regions. Now, I'm not talking about patients, I'm talking about regions, okay, lung regions identified by CT scan. Percent of regions that became PA negative at 1.5 years after ETI, and what we saw was the identical rates. The highest regions, about 40% cleared. The lowest damaged regions, same thing. When you look by change in Pseudomonas CFU counts, you see a similar pattern. Both the high and the low experienced about the same 50-fold or close to 100-fold reduction, more like 50, in Pseudomonas CFU. So it does not appear to be persistent infection only caused by the severely damaged regions remaining a nidus for infection. So the last question I'm going to try to address is, how does ETI affect lung inflammation? And I'm going to be showing you data from the bronchoscopy study of Sam Durfee. And I'm going to be focusing primarily on one marker, neutrophil elastase. I think this is our best marker of inflammation in CF. There's been a lot of good epidemiological studies that shows that neutrophil elastase levels predict the progression of lung disease and bad outcomes in CF. And it's also a very good marker in non-CF bronchiectasis, so it might be particularly relevant to the post-ETI people. So going back now to our bronchoscopy study, sampling the 2 worst and 5 best regions in each person's lung, and I'm going to show you the neutrophil elastase data by region. Okay, so on the y-axis is plotted the neutrophil elastase level in these lung washes. Each point is a region, not a patient, before and after 1.5 years of ETI. And what you can see is a really marked decline. Interestingly, I'll come back to this in a second, a bunch of patients became neutrophil elastase undetectable after treatment. Now I'm going to show you this data in 2 more ways because we thought it was really informative to look at it in different ways. First, I'm going to show you this data based on the status of whether the lung region had a persistent PA infection or whether PA cleared. Same data, just displayed differently. So these are the PA persisted patients. These are the PA cleared. Sorry, I said it again, patient. These are the regions, 5 per person, PA persisted, PA cleared. And what you can see is that in the PA cleared regions, neutrophil elastase in all but one case became undetectable 1.5 years after ETI. I want to show you this data one more time a slightly different way. Now I'm going to show you the relationship between PA on a log scale detected in each region and the neutrophil elastase level in that region, okay? Each dot represents that relationship for one particular region. This is the pre-ETI data, okay? Saw a nice correlation, R-squared value of 0.28. It was statistically significant. What we noticed post-ETI I thought was striking. First of all, the total neutrophil elastase levels are coming down. And secondly, what you can see is that there's a much stronger correlation, R value is now 0.76, between PA level and NE level, okay? A better goodness of fit. So for every log unit of PA, better predicts the log unit of NE present in that lung region after ETI. And this, I think, has some interesting implications. So when we think about inflammation in CF, there's probably multiple mechanisms that cause a lung segment to be inflamed. There's pro-inflammatory effects of the CFTR mutation itself. Mucus plugging itself, even without infection, is postulated to produce tissue hypoxia and marked inflammation. And of course, there's the effects of the bacteria producing toxins and inducing toxins from the host. All these factors induce lung inflammation in CF. Now, our data showing that the goodness of fit between PA and NE markedly improves post-ETI raises the possibility that some of these other inputs have been diminished in importance. ETI did a good job, perhaps, in reducing these inputs, but this input, the toxins produced by the CF pathogens, may remain the key driver of inflammation in the post-ETI era. So my last slide just going to show you that this dichotomy in the change in inflammation in the PA persisted regions versus the PA cleared regions was true for other inflammatory markers as well. Here's a panel of 13 that are shown. So to conclude there, our data suggests that ETA markedly reduces CF pathogens in sputum, but most patients remain persistently infected after treatment. ETI did not change the absolute abundance of the anaerobic species detected in sputum. I think that's likely because most of those are coming from the mouth, from the saliva as contaminants. Post-modulator infections may not be a consequence of colonization or infectionitis of the most highly damaged regions of the lung. We found an all or none phenomenon. If a region cleared, usually all the regions in that subject cleared. And so it does not appear to be this explanation. I want to caution that this is a small study. Neutrophil elastase levels become undetectable in lung regions that cleared P. aeruginosa. Again, important, but caveated with a small study warning here. And our data looking at the goodness of fit between inflammation and PA suggests that persistent pathogen infection might be the key driver of lung inflammation in the post-ETI treated patient. Again, that was a small study. So the PROMISE study had many contributors. I want to point out Dave Nichols and Sarah Morgan. Dave was my co-PI, and Sarah did a lot of the work, as did Peter Jorth and our statistical colleagues and collaborators. The bronchoscopy study was primarily led by Samantha Durfee, a PhD student, and it was a collaboration between us and investigators at the University of Iowa and also at Cincinnati. So thanks a lot for your attention. Happy to take questions now or later. Yeah.
I think we can probably do a question or two now. Okay, so reminder for folks to put your questions in through the app. Let's see, our top scorer from Rachel Rayner. What types of non-CF microorganisms are detected unchanging in the sputum, and how does this compare to people without CF? I mean, would you consider these airway commensals or?
Boy, that's a tough question. I think sampling the normal airway is extremely difficult because the biomass is so low in a normal, healthy airway, and because the airway sits behind a extremely high biomass region, which is the mouth. The concentrations of oral bugs in the saliva is 10 to the 9th. Some people find 10 to the 10th. Edith Semantic's work suggests that. So even minute amounts of contamination can give you a signal. Imagine if a milliliter of sputum was contaminated with a microliter of saliva. That could give a signal of 10 to the 6th organisms just from that contamination. So it's a difficult question. I think Robert Dixon has done good work in that area, and he finds that in the normal airway, bacteria are deposited but are unlikely to be growing or replicating in the airway, that they're likely cleared by normal host defenses.
Great, and we'll do one more question and then we'll save the rest for our panel at the end. Sebastian asked, what do you think comes first? I think this is on everybody's mind. Does ETI reduce some of these other factors, other drivers of inflammation? This reduces the susceptibility, improves the host response, or do you think it's the opposite? It's really directly working on the pathogen burden, and this is then reducing the immune response and neutrophil activity? Thanks.
That's a tough question to answer. Our data does not distinguish between those possibilities. Both are formally consistent. Both possibilities are consistent. I would say, though, that a predominance of other data suggests that infection may be a consequence of immune dysfunction rather than immune dysfunction being a consequence of infection. Both could happen, but for example, when you lower immune defenses in an animal, you get spontaneous infections. And there's much less data suggesting the opposite, that increasing the activity of immune responses, increasing inflammation will produce inflammation. But I don't think that's answerable in a human, at least this human study.
Yeah. Great. Thank you so much.
Sorry, if you see us looking at our phones, it's not because we're being ignorant and looking at the news, although there's always news at the moment, isn't there? So next, it's because we're checking the questions. So next talk is going to be by Dr. Robert Quinn, who is with us from Michigan State University. And he's going to be talking about the modeling and testing the future of the CF lung microbiome in the era of the highly effective CFTR modulators. A bit of a mouthful, but he also told us if he finishes early, he's going to give us a dance. Not quite sure what that means. So be prepared is all I'm going to say.
Yeah, I shortened it to tricafta, but I've been practicing saying lexicaptor, kesicaptor, ivacaptor, so maybe that shortens it. Anyway, thanks guys for the invitation to speak today. This is a really important session. We're going to talk about the work that we've done on the lung microbiome to understand the changes that are happening with this revolutionary medicine, tricafta, that's had such a beneficial, exciting impact on the lives of people with CF. And throughout the talk, I'm going to show art by an artist named Dylan Mortar. He's a person with CF, and he depicts, in this image in particular, his experience with his disease. And I think it's a good kind of thinking point about what we think of CF in the past. What will it be like in the future with this highly effective modulator and maybe even better drugs to come afterwards? I change the slide. Okay. So I have no conflicts of interest and nothing to declare, except that please visit Dylan's website. So Dylan is actually here at the conference. He can't be physically present in the building, of course, but he's here. Him and I developed a bit of a relationship recently. He visited Michigan State a few weeks ago, and I believe he's at the Iron Hill Brewery, kind of hanging out if you guys want to talk with him, which sounds like a great place to spend 3 full days. So I've been studying CF now for about 10 years, and I actually did my PhD studying infections of lobsters and wrote a postdoctoral fellowship for the CFRI, which is a great nonprofit institute, to study CF exacerbations about 10 years ago. And when doing that, I remember reading a lot about these mucus-plugged bronchioles, okay? And this is a histological section showing just that, with the mucus in blue stained in the middle. And I trained as a microbial ecologist, so thinking about that system, this is how we think it sets up. So we have thin bronchioles in the airways plugged with mucus. And in that mucus are bacteria. Relatively more diverse system than we originally thought. There's many different types of organisms there. And as sort of first principles, we can actually predict certain things will happen. So oxygen gradients that we talked about, my lab has been studying this in vitro, looking at how the community stratifies in the presence of chemical gradients for quite some time. And actually, first paper I published was back at San Diego State University with Forrest Rower in 2014. Working with Katrina Whiteson, we basically described the metagenomic pathways that exist, the core pathways of the organisms that survive there and allows them to grow and persist. The question now is what's going to happen with this revolutionary therapy, tricafta. So as it begins to penetrate into the airways, are the bacteria going to go away? Will their pathways go with them? Will these oxygen gradients begin to reverse? And is this the situation that we're studying now? I think Pradeep is showing that there's been drastic reductions in all of the bad things about CF, but not complete removal. But I'm not a clinician, so I don't have the inference that many of you have. But we're really trying to understand what this system looks like now. And for a bit of historical context, so we've thought a lot about and written periodically about microbial ecology in CF. And we have this, what we call the climax and attack model. Really thinking of these two groups of organisms as guilds, okay? So the group on the left is called the attack community, and the one on the right is the climax. Attack community being this anaerobic bacteria that ferment sugar, lower the pH of the environment. The climax community is the antibiotic-resistant pathogens, organisms like Pseudomonas aeruginosa. They have fundamentally different carbon sources, which is important for the rest of this talk. We believe, and there's been a lot of evidence in the literature really for decades, that Pseudomonas aeruginosa prefers amino acids as its primary carbon source. So a paper we published a few years ago, we did a cross-sectional study. I'm going to talk a lot about metabolomics today too. So my lab does a lot of metabolomic profiling, untargeted profiling of small molecules in many different samples, sputum being one of them. What we found in a metabolome study in 2019 is that there's a real association, and Pradeep really touched on it as well, kind of reversing, in inflammation in the climax community, or these antibiotic-resistant pathogens. So in the airways, the presence of these organisms induces massive neutrophil recruitment, okay, massive amounts of neutrophils, and they bring with them their antimicrobial properties, one of the most important being neutrophil elastase. So in our metabolomic data, we asked a simple question: what are the molecules most associated with poor lung function in people with CF? And the answer was peptides. People that have poor lung function, their sputum is absolutely loaded with peptides and amino acids, okay, which we think has a particularly important relationship with Pseudomonas aeruginosa. So this organism that's able to sort of persist the inflammatory attack is essentially bathing in its primary carbon source due to that inflammation itself. But we keep asking the question, is this starting to reverse? Will Trikafta have an effect? Now, I'm not the person to tell you about the miraculous effects of Trikafta and the symptoms of CF, but we all know it's had really positive, in some cases almost unexpected impact on their symptoms. One of the impacts that has been somewhat challenging for people like myself is the absence of sputum production. So people with CF that are on Trikafta have more difficulty producing sputum, which makes it difficult to study the microbiome, and Pradeep mentioned some alternative ways to sample. So what we're doing then is trying to get to some ground truths using mathematical modeling. So how can we predict, understanding, or using what we think we know about this system, what the future of CF microbiome might look like? This is work I've done with Chanyu Zhang and Isaac Clapper, who is at Temple here in Philly. I think he may be at the conference today. So these slides, basically what I'm showing is sort of a epithelium on the bottom. So imagine this being a bronchiole, the lower airways to the left, and then the upper airways to the right. Sorry. And we'll look at some of these model predictions of the mucus layer. So the pre-Trikafta example, when we have slow mucociliary clearance, we believe that the mucus kind of builds on its way out, right? So the thickness of the mucus increases, becomes very thick the further you move towards the mucociliary clearance. What we're showing here is the oxygen profile. So oxygen penetration really only goes about 2 micrometers into this layer. And there's a lot of anoxia predicted in this system. So in this model, we also have our two guilds, represented by Pseudomonas in this case, and these anaerobes. Our model predicts that Pseudomonas can really thrive everywhere. It's a facultative anaerobe. It does well aerobically and anaerobically. But not surprisingly, the anaerobes this model predicts really don't do well in the thinner layers where the oxygen penetrates and have a niche space deeper in the mucus. And we've been studying this for quite some time, and that certainly appears to be true. So again, the question being, what happens now that this triple therapy is present in this system? So what we basically modeled was an increase in mucociliary clearance. So things are moving out faster. And the result, the model predicts a thinner mucus layer. Oxygen penetrates further into the mucus in this case, basically the entire layer. So we predict in this case, Pseudomonas will persist. It's able to find its niche space, it tolerates oxygen, but the anaerobes are running out of places to grow because the mucus layer is becoming thinner, and this is what our model predicts. I don't think I'm totally giving it justice. In some ways, we probably could have came up with that over a beer, but this model is actually far more complex than it seems. There's a lot of parameters incorporated here, including growth rate of organisms, biofilm structure, oxygen penetration, interactions between the organisms we think we know from experimental data. And I want to make sure I give Isaac and Chanyu their justice. So we've done a study we published last year, the first look at the effect of ETI, Trikafta, on the lung microbiome and metabolome. Relatively small study, so we got paired sputum samples before and after a subject took Trikafta. And we're looking here, these box plots, at the changes in the alpha diversity from the microbiome and the metabolome. So I'll highlight a few important points. You can see on the left the Shannon index, so the diversity of the community is increasing. And that seems to be driven primarily by an increase in evenness. So the community is becoming more even in their relative distributions among the organisms. A similar thing was seen by a group on the previous modulator as well. And then in the metabolome, now full disclosure, we're not totally sure what diversity of a metabolome necessarily means, but what we see is the total number of molecules we detect in the sputum when they're on the drug has gone down. So we're losing chemistry in a way. Again, principal coordinate analysis plots that you saw in the previous talk, these are basically ways of comparing the similarity of this high-dimensional data. Each sample is a sputum sample colored by pre and post ETI. The thing about this, and it's a real kind of issue in CF microbiome research, everyone's different from each other, right? So they all start in different places in this plot. What we're able to characterize in that first axis is they have a shift in the similar direction in both the microbiome and the metabolome. So the drug is causing a change in their lung, and they all seem to be changing in a particular way. Another thing that we characterized was looking at the Curtis distance, so the amount of difference among the metabolome in particular before and after ETI. What we find here is that when people are on the drug, their lung metabolome goes much more different from each other. So there's relative similarity within the metabolome prior to the drug, and then when they're on it, we had this kind of what we described as metabolic chaos. We don't totally know what it means, but we saw that. And then looking at things we know we care about. So what we saw in this study was that peptides and amino acids are decreasing in their abundance in this sputum, which we think is important again for Pseudomonas. Tryptophan, for example, decreases in its abundance, and even pathways associated with amino acid metabolism, such as kynurenine, are decreasing in the sputum when people take Trikafta. Looking more at things we know we care about, so the total bacterial load, again, we had a small sample size here of about 24 people. We saw a small reduction in the total bacterial load. It didn't reach significance in this case, but certainly trended so. And then the relative abundance of Pseudomonas in this first sample goes down, but we did not reach significance in this case. Pradeep showed with higher sample sizes that a massive reduction. And you can see lots of people have decrease in Pseudomonas. Some people it actually went up. And again, this is the relative abundance in the microbiome. So I really advocate for those of you that are working or trying to work on microbiome data to do these log ratio kind of binning of organisms. So if we bin the microbiome data into our two guilds, the pathogens and anaerobes, compare the log ratio of pathogen over anaerobe, now we get our statistical significance. See, because not everyone has the same pathogen, right? So you get a pretty small sample size if you start dividing it up. We see a significant reduction in the amount of pathogens relative to anaerobes on Trikafta. So what we kind of argued, or at least proposed in this study, is that Trikafta is reshaping the niche space for the climax community, or the antibiotic resistance pathogens. We're kind of seeing a reverse of what we believe was a kind of common characteristic of this disease. The peptides are going away because neutrophil elastase and the inflammatory signals are being reduced, although we didn't measure inflammation in this study. And again, what might this mean for Pseudomonas aeruginosa? So we've been playing with some other models and even kind of just thinking about what might be happening. We could predict a possible crash in the entire system, the microbiological system, where the oxygen penetrates too far so the anaerobes can't survive, and if the primary carbon source of Pseudomonas is also going away, the organism may not be able to survive itself. That crash may have already happened. The fact that people can't produce sputum so much, Pradeep showed massive reductions in the total bacterial counts. This may be why. So the last thing that we've been doing, we call the freezer project. This is kind of inspired by John LaPuma's work, where we basically ask people with CF, instead of throwing your sputum sample in the garbage can, how about put this little freezer in your house and you can just throw it in the freezer and we'll come pick it up from you after a year. So this was published in 2020. This PKA plot of the metabolome kind of gives you a picture of the sample size. So we collected 615 sputum samples from 6 patients over a year and did this multi-omics 16S microbiome and LC-MS metabolomics. And when we look at the microbiome data, this really shows you the difference in the communities among individuals. Okay, so we're colored by person here. You can see the Stenotrophomonas patient at the top left has a very different microbiome from the Pseudomonas group and even an Escherichia patient that we've been studied who has a unique mutation. So what I'll show now is an animation, assuming that we can get it to work, of the movement through PKA space. And these are basically daily samples in the freezers of patients in their homes. What you can see is the microbiome in each person is kind of moving through this PKA space in a particular direction, kind of back and forth through time. We actually characterize this, and it appears to be driven by these changes between the pathogens and anaerobes, sometimes driven by antibiotics, but not always. Trouble with the, okay. Oh, just played it again. How do I slip the next slide? There. So now we're working on Freezer Project 2.0. Okay, so now that people are on Trikafta, This is an important point. Because of the difficulty in getting a sputum sample for people on Trikafta, it made sense to us to put freezers in their homes. They can more opportunistically produce a sample than they could perhaps on demand in the clinic. So this image kind of shows the longitudinal sampling scheme. We have a pre-Trikafta group, which actually includes the previous study, so we're comparing to the previous data. But we have 8 individuals with freezers in their homes, and they're collecting through time. You can see we have a decent sample size for each individual, around 150 days on average. But you can see the little arrowhead point is the point when people began taking the drug. So it's not a perfect study design. The pandemic did not help this. But regardless, we have a pretty reasonable look at the dynamics similarly to the previous study. And here's that PKA plot. So right away, what you can see is some individuals have a microbiome that really maps just with people that had a pre-Trikafta situation. They don't look all that different. But if we watch the animation this time with this blue group on Trikafta, it's a little bit of a subjective with your eye, but I think you can see that the directionality of the change is sort of lost. It appears to be a little more chaotic movement through this PKA space, and we're actually able to quantify that. Keep rerunning the animation. There we go. So here we have these box plots that are characterizing the change of this microbiome and metabolome in this longitudinal study. So the top is the alpha diversity and the bottom is the beta diversity. Again, beta diversity is a fancy word for kind of difference among the microbiome. On the left, we have the Shannon index. So you can see this is normalized per day. So the time between samples, the change in the alpha diversity, time between samples per individual. Basically, how fast it's changing. And what we see is that on Trikafta, the alpha diversity is changing more per day than it was prior to the drug. So things are moving quicker. The beta diversity, the change in the difference in the microbiome through time, is faster again. So when they're on the drug, and this is true for both the microbiome and the metabolome. So the microbiology and the chemistry is changing faster while they're on Trikafta. And I should mention, we're very aware of the fact that the subjects we're studying are still producing sputum. We're not sure if there's somehow a unique population of the Trikafta subjects who, many of them, do not readily produce sputum. And one thing we found is that they're still very much infected. So the microbiome, and this is relative abundance, but the microbiome data here, these don't look all that different than what I'm used to seeing. So the blue is Pseudomonas, so 4 of the patients have really dominated by pathogens. Burkholderia is in orange. So through time, these people are still very much infected. We understand their lung function has improved. They're still producing sputum, but things are changing faster. So that brings me back to this image, right, that I think a lot about. And I wonder what the experience of people with CF, perhaps future artists with CF, may depict. Will this be the traditional experience? I'm not sure. Things are certainly looking up, looking positive, and we're seeing changes in the lung microbiome that reflect that this drug is having a major effect on that microbial ecology. So I'll say thank you to many people. I work with Spectrum Health at Michigan State, University. They've been a great group to work with, as well as Doug Conrad at UC San Diego. A number of members of my lab, and I'll thank Dylan for letting me kind of hijack his art for the last few years.
Great. Thank you, Dr. Quinn. That was an excellent talk. I'm not sure I understood a lot of it.
Okay, that's not good. That's my fault.
But no, it's incredible science, so
thank you.
Brilliant stuff. So we have one question at this point from Meti Kolpin, and that question is, when the mucus layer becomes thinner, there is probably still the possibility of oxygen-free zones as the neutrophils consume oxygen by the respiratory burst. Have you measured oxygen and mucus before and after ETI treatment?
No, we haven't. That would be a great thing to do. We're also interested in the pH, how the pH might have changed. Yeah, I think just our model predicts, oxygen penetrates constantly, right? Because it's in the air, obviously. The thinner mucus, we can ask Isaac about that, but generally the diffusion rate of things is faster when the mucus is thinner. So it may very well be that a lot of the anoxic regions are gone, but the impact of neutrophils, I really don't know much about that. I couldn't comment, but if they're respiring oxygen, that may also reduce it. But again, if the neutrophils are not being recruited to the amount that they were before, even that might be going away.
Can I just ask an additional, can you see a clinical utility of any of these tests? Any way of getting this information into something that's easily digestible for clinicians?
Yeah, absolutely. We tried to do that for a long time, back when I was a postdoc in San Diego. So we actually published a paper a few years ago that we could actually generate, produce, and analyze this data in 48 hours. Particularly the metabolome. I could pretty much tell you if Pseudomonas aeruginosa metabolites are present in a sputum sample within hours of its collection. But the challenge is the interpretation. So physicians' decision-making is a complex process of which bug to kill. I don't know how to tell someone which bug to kill. We can tell them what's there, we can do it in 48 hours, but the interpretation of that data from a clinical perspective is still challenging.
Excellent. Okay, so we'll have more discussion on this, I'm sure, at the end. So we're just going to go to our next speaker. So thank you. So our next speaker is Mimi Saavedra, and she's going to talk to us about T cells. I always get very worried when people mention T cells because it means we're talking about immunology and complicated immunology. So, and it's something we've probably not spent enough time thinking about in CF is the adaptive immune response. So really looking forward to this talk.
Thank you. Okay. Thank you so much for inviting me to be a part of this. I'm really honored to be talking at the symposium. I'm actually also pretty excited to have a talk that's got the word hangry in the title. I'm going to talk to you about the effects of Trikafta on host immune cells, but like was just mentioned, it's probably the immune cell that people are least likely to think of when they think of the CF immune response, and that is the hangry T cell. Okay, great. All right, I have no disclosures. Okay, why T cells? So my interest in T cells arises from an observation that has come to me in many years of taking care of patients, and that has to do with the geography of pulmonary exacerbations. And so this is one of my patients at her baseline in January of 2021. So these are all post-modulator pictures. And you can see that when she experiences an exacerbation 6 months earlier, that she's mainly affected in her lower lobes. And then 1 year later, when she experiences an exacerbation, again, she's mainly affected in her lower lobes. And in years of just looking at x-rays and looking at the progression of disease and what's happening during the exacerbation, I've always been struck by why are the infections always happening in the same place, same species, even same strain of pathogen, and what's the adaptive immune response doing in these patients during those times? Because what actually should be happening is that when she first was infected, her naive T cells went ahead and proliferated, differentiated, created effector T cells, which would be around on the order of weeks, days to weeks. And then also this population of memory cells, which should be long-lived population, so that next time that she got sick, she would have, especially with the same pathogen, she would have this much more robust secondary response in which she would have a rapid induction of effector function. It would be way more effective, way more fast than it was the first time. And that's actually the principle of why when we get an infection, we generally don't acquire the exact same infection again, and it's the whole principle behind vaccine-related immunity. So when this happens, this secondary response, these memory cells have to make a very rapid metabolic switch. They need to eat, and they need it immediately in order to be able to produce this very quick and efficient effector response. And that had a lot to do with the hypothesis for this study, and that was that CFTR is very important for healthy metabolism, that T cells were very, very, very dependent upon this rapid metabolic switch so they could have swift recall and swift effector function. and that Trikafta would basically create CFTR rescue. So what we hypothesized is that memory cell metabolism genes would change in a way that would help support why patients have reduced pulmonary exacerbations, not just from the microbial side, but also from the host side as well. So the way that we did this is very quickly, we mobilized this study because, as you all know, nobody knew when Trikafta was going to be approved. So right when it was approved, we were able to get samples from 20 patients before, and then about 4 months after, we took their peripheral blood T cells. Why peripheral blood? Well, remember, memory cells are by definition antigen experienced. There's a rich population of memory cells in the circulation. And these cells, they traffic to the lung, to the sinuses, to the GI tract. They come back into the circulation where they're directly evaluable. The technique that we use is called site-seq, and with site-seq, you're able to do simultaneous protein expression and single-cell RNA sequencing at the same time, and that's very important. I'll get into that a little bit more. But basically what's happening is that you have your single cell, you have an antibody so that you can define what that cell is, and then conjugated to that antibody is a barcoded oligonucleotide. And so what happens then is that you get differential expression of genes as expected from the single cell, but you're able to very precisely identify those cells based on the ADTs. We also additionally did plasma metabolomics and CD3 T cell metabolomics, but I don't really have enough time to get into that, but there is some more kind of beyond this story. One of the things that was really important to us wasn't just to say, okay, what's the delta? What's the differential expression of genes before and after Trikafta? But really what we wanted to get into is this association between cell type specific gene expression and these really important clinical outcomes with improvements in FEV1 and reductions in hospitalizations. And all this work was done using a generalized linear mixed model framework, which was very elegantly done by our brilliant computer scientist, Brian Vestal. So two quick things I want to mention from the approach is that basically what you end up with are antibody oligos and then mRNA oligos. Both of those have library builds and both get sequenced. And then when that happens, you will have a pool of data that's basically your single-cell RNA-seq data, and then you have your ADT data. And what's nice about that is then you can get differential expression of proteins, but also when you're making your TISD plots or your UMAPs, that all these individual clusters, you're not depending on the gene expression to define what those cell types are. You actually know what those cell types are. And so when we did this experiment, we basically had 9 CD3 T cells types identified from the peripheral blood. And this is so beautifully, using this technology, laid out. And I actually have to give compliments to the people who did these experiments, did a beautiful job. But you can see here that there's a CD4 population here, right? There's a CD8 population there, and that includes CD4 tissue-resident memory cells and CD8 tissue-resident memory cells. There is, on the bottom of this UMAP, CD45RA positive, so those are the naive Treg, CD4, and CD8 cells. Then we use CD45RO as our memory marker so that you can see CD4 memory, CD8 memory, tissue-resident memory cells, Treg memory, and then we use CD25 for the Tregs. And this little kind of bridge between CD4s and CD8s were the NK population. Now what you can see here with all these different cell types in parentheses are the number of genes that were differentially regulated before and after Trikafta. And what I'd like to point out is that the CD4 group actually had the most number of genes in general, naive in memory, changed after Trikafta. And amongst the memory, the CD4 memory had the most number of genes amongst all the memory populations. Where's my pointer? Oh, there. Okay, a thing that I'd like to just go back to is just visualization. And visualization is incredibly important when you think about this data and single cell data in general. So a T cell is not just a circle with a huge nucleus. It's more like a sunflower. So it has all these microvilli projections, and TCR is basically not preferentially on the body of the cell, but it's in those microvilli projections. And the reason for that is that they're looking for MHC complexes on antigen-presenting cells so that they can bind, and when they bind, that's a prolonged contact that needs to happen in order for full activation to occur. And that site where that's happening is the immunological synapse. And so in that immunological synapse, once that happens, then there are then signals for chemokines to get upregulated into that synapse, so then they can create these appropriate co-stimulatory signals, because you want that antigen recognition to occur and then T cells to migrate to that site. And that is really beautifully shown here on the CyTOF data. So you can actually see, here's a chemokine CCR2 population of CD4 cells there that you could then identify the expression of those genes. CCR5, which is important for reducing glycolysis in memory cells, so it's a regulator of metabolic fitness, and you can see its expression here on this kind of peripheral portion of CD4 memory, CD8 memory, and then the most highly expressed CXCR3, which is important for TH1, and then you can see that's kind of distributed throughout both the CD4, the TRMs, and the CD8 memory populations. So if you understand the biology, you can really use this kind of technology and take advantage of what's actually happening. The other thing that you can do that's really nice is that you can say, okay, well, for each of those groups, what are the primary markers that are gene markers that mark those groups? And so here you can see CD4 memory, right? And here's the primary markers, so C-type lectins, S100 proteins important, genes important for chemotaxis, annexin A, which is anti-inflammatory, and you can see that the CD4 memory and the CD4 tissue resident memory, so what's actually happening at the level of the tissues, that those markers are similar. But when you look at CD8 memory, then the markers that are happening in those peripheral circulating CD8 cells, so the granzymes, are not necessarily what's happening at the level of the tissue resident memory in the lungs and the tissues where those are present, and actually they're a little bit more similar to the NKT markers. And so that's something that's very useful when you're trying to analyze this monster of data that comes from these experiments. Now back to our cohort. So these were 20 patients, and the thing that I would like to emphasize is that there was heterogeneity in the change in their pulmonary exacerbation rate in the year before and the year after they started taking meds. And then there was also a lot of variation in their FEV1 response, right? They're humans, so this is completely understandable. Instead of going into the differential expression of genes before and after Trikafta, I really want to focus on Brian's work with his mixed models, looking at how does a change in expression for each given gene within one person affect the number of hospitalizations or affect the improvement of FEV1 within that person? And so the CD3 T cell memory group that had the greatest number of genes for hospitalizations and for FEV1 was the CD4 group. And this number is how many genes were significantly changed in relation to the change in hospitalizations. And these are heat maps that Brian created. Now, on the columns are the patients, and then on the rows are the genes. And what is so interesting is this is where he basically gave you an idea of what was happening with the FEV1 along the top. And so he called the green FEV1 responders. He called the FEV1 non-responders. Now, the FEV1 non-responders generally had FEV1 improvements of less than 5 absolute percentage points in their percent predicted after starting Trikafta. And the FEV1 responders had over 5, though a lot of these people had it into the teens and into the 20s. So they really responded. And so while you see kind of this bland color over on the side of the non-responders, the responders who really had FEV1 improvements had this biphasic improvement, which we'll actually see as a recurring theme in our hospitalization data too. And so on the top, the first 3 genes actually are related to mitochondria, and the first 2 are related to complex 1 of the electron transport chain. And then on the bottom, there are a variety of genes, but there are definitely represented here groups of genes that are related to autophagy. So these people increase those genes within their mitochondria to help encourage cellular respiration and reduce autophagy genes. It's really nice when you have a variation in responders because that really drives your heat maps and your data. And so this is just going back to the biology. If you remember when we talked about memory at the beginning, is that when a cell is going to be activated and it's a T cell and it needs that metabolic demand to be there and ready to go immediately, the currency that it needs is ATP. And so those first 2 genes basically came from that heat map that were so red, came from complex 1 of the electron transport chain, which is the NADHD hydrogenase. So NADH gets oxidized, generating a hydrogen ion, which eventually contributes to this hydrogen ion proton gradient that will then lead to the production of ATP. And ATP is what they need in order to switch memory effector, and be able to do that in a very robust fashion. And so what about hospitalizations? So when Brian did the Poisson analysis for hospitalizations and expression of genes, he identified that out of those 9 groups, 5 of those groups were significantly associated with hospitalizations. These are the 5 groups. And what he also showed on this Venn diagram is that there were genes that were in common to the whole CD3 lineage, so that would be these 151, but then there were also genes that were very specific to the cell subtypes. And so that, as an example, CD4 memory and CD4 naive, this combination of a CD4 group had the greatest amount. And so those were the unique ones to those particular cell subtypes. And so showing you the same heat map, again, people had a lot of variety. And so here are the columns of patients and here are the rows of genes. And what you can see here is this actually, the blue are patients who in the year before they started Trikafta had 0 exacerbations, and the year after they started Trikafta they had 0 exacerbations. So they were just kind of flat, right? And that's a kind of a unique group, and you can see that kind of flatness in the color. But then to this side, there were patients who went from 6 hospitalizations in the year prior to 0, or 5 to 0, or 4 to 0, and that group really drove kind of the expression patterns on this heat map. There were folks who increased their hospitalizations after Trikafta, and you can see the difference, right? And so when you look at these 2 groups, one of the things that we were able to do in the analysis is that we were able to say, okay, per 10% increase in expression of the genes, how would the hospitalizations change? And so when you look here, a 10% increase in YTHDF1 led to an 85% increase in risk of hospitalizations. And when you look at the particular genes here in this group, you have this one, which has to do with RNA methylation and glycolysis, this one related to glycolysis, this one related to NF-kappa B stimulation, and this one actually related to cytoskeletal arrangements. Think back to those T-cell electron micrographs, right? And all the arrangements that need to be made when they're actively recruiting and getting into those sites once they've made that MHC connection. The interesting thing is that this also is involved in cytoskeletal arrangements, and yet this is a gene where a 10% increase in that gene leads to a 47% reduction in hospitalizations. And these other ones tend to have anti-inflammatory effects, and this is a mitochondrial protein. So you can definitely see how these patients who had marked changes in hospitalizations actually had different genes that helped them in the CD3 T cells compared to the ones that helped them improve their FEV1. Those were looking at individual genes. We also wanted to look at network analysis so that we could look at groups of genes that might fit into biological pathways that would then correlate with change in hospitalizations after starting on Trikafta. And what we used was WGCNA, and so this is a module that came out highly significant. This is the Emmy Blue module. It's for the CD4 memory group. And what you can see in this group is that the regression coefficient is positive. That means that an increase in these genes was positively correlated to an increase in hospitalizations. Now, this particular gene is the hub gene. That's the one that had the most correlation with the other genes in the module. And this gene is actually NR4A2, which is a nuclear receptor which promotes differentiation to Th17. That's probably the biggest thing that's been described in terms of T cells and CF is Th17 biology by multiple groups, J. Coles, Tyringer, Mosley, Broadley, and so this actually makes sense. And so a 10% increase in this hub gene leads to a 50% increase in hospitalizations. When you looked at the WGCNA, another highly significant group that had negative regression coefficient was the light cyan group. And so in that group, an increase in these genes led to a decrease in hospitalizations, and lo and behold, the hub gene for that group also makes up one of the subunits for complex 1 in the electron transport chain. And so a 10% increase in this leads to a 40% decrease in hospitalization. So this is a whole other way of looking at it, network analysis, but showing similar data to what we saw with the individual genes. So this goes back to chicken and egg, right? This is what always happens with CF research, especially when you're doing it on humans. And that is, well, these people have better lung function, they have less infections, they have less exacerbations, their nutrition's better, they weigh more. Of course, their immune cells are going to look different and work differently. Yes, absolutely. And our plasma metabolomics work definitely supports how much metabolic change is happening across the whole individual and also in their individual cell types. But then there's the egg, right? And that is everything a secondary effect that happens in the immune system? Could some of those be a primary? And this is just basically the end is the beginning, right? So this is towards the end of my talk, but the beginning, and also this issue of thinking of what about CFTR in the individual immune cells? And this is just a plain Western blot. It supports data that's been published by multiple groups, and basically showing that in CD3 cells, CFTR is actually expressed. And so it is possible that distinct processes are happening within CD4 or CD3 T cells in general that change some sort of aberrant metabolic programming that may ultimately help their function. And that takes me back to the hangry immune cell phenotype. I really like this figure. It's from a European Respiratory Journal review, because what it's really pointing out is that when we think of CFTR, we shouldn't just think of it in terms of all the changes that happen at the plasma membrane, but also to consider everything that happens in the intercellular dynamics when that happens. And that's at the level of multiple organelles, including the mitochondria. It's that increased metabolic stress that occurs when you have these huge protein complexes that require more metabolic energy to, basically take care of them. And so the issue I think that comes out, or the conclusion that most prominently comes out from these analyses, is that the energy that it takes to remember an infection, to have that adaptive memory, may be more efficiently propagated, empowering this process in general in the presence of Trikafta, probably secondary effect, maybe some primary effect. The questions that arise from that is that heterogeneity. When I showed you that heterogeneity of FEV1 response, or the heterogeneity of hospitalization response, can some of that be traceable back to what the immune cells are doing? And we have all these new categories of agents, right? 6 or 8 now that are kind of coming into clinical trial. Can they augment CFTR function better? What will they do in terms of these processes in immune cells and helping them to remember and helping them to deal with infections better? Okay, in conclusion, we have a wonderful group who worked on this project. The people who actually did the single cell, which was a very expensive one-time shot, did an incredible job. Katie and Sylvia went to all the patients, collected. The patients were incredibly gracious. They wanted to get on this drug ASAP. They waited till we could time the draws so that we could get the single cell done because it was all done fresh. They were incredibly great about that. We had great PhD input in terms of what was actually happening from our data, and Brian is an incredible scientist. His data actually is being presented throughout this entire conference. Thank you.
That was an amazing talk, Dr. Saavedra, and I kind of like T cells now after that. So we've got maybe one question before we take the rest of the questions to the discussion, and this is from Ajay Dandekar, and their question is there a transcriptomic signal of T cell exhaustion before ETI, and is there evidence that it is reversed after ETI?
Yes, there absolutely is. Prior to ETI, we had actually done tons of flow on patients at exacerbation. So I'm going to actually jump to our older data, and on that flow data, it was incredible how much PD-1 is expressed by those cells in patients before, like right when they're coming in, right when you're starting to treat the exacerbation, right? So that time when that metabolic switch should be kicked in and those effector cells are pumping out, those exhaustion markers were huge at that specific time where they needed them the most, suggesting, that aberrant metabolic function is leading to dysfunction in memory response. So obviously, that's not the most important response of the CF immune response to infections, but it's a component of it that doesn't seem to be working well. And yes, those PD-1, TIGIT, all of those seem to improve in the patients following treatment.
Great, thank you.
All right, now we're going to conclude with our last talk and remind you that we'll have a panel discussion afterwards. So I'm going to invite up Robert Gray, my co-chair, to finish this off and kind of talking about resolution and repair and inflammation.
Okay.
Technology is fun.
Okay, great. Well, it's great to be here and really delighted to be giving a talk in this session to round off the more formal part before we have a discussion which will probably involve myself and Ben manoeuvring some furniture around and trying to make things a bit more interactive. So I'm going to talk about inflammation, resolution and repair in CF following modulator therapy. And when I looked at the title over the last couple of days, I did realise it was probably a bit of a big subject to fit into 20 minutes. But we're going to have a go anyway. So let's just move on to the next slide. So these are my disclosures. And yeah, let's talk about CF inflammation to start off with. So the CF airway is, or possibly was, but most likely is, still an inflamed environment. This is a figure from Dave Nichols' review on CF airway inflammation published in 2015, and he calls this the simplified view of the CF airway inflammatory response. And I think if Dr. Nichols is here, we have to ask him what was the complicated view of the CF airway inflammatory response, because it must have been terrifying. But what it does show us is that inflammation is really complicated. And we probably don't understand how all these different facets of inflammation interact with each other. And that's probably manifest in the detail that we do not have particularly good anti-inflammatories to use in our CF patients. A more simplified view, perhaps, is to think of the way that the sort of inflammation I'm going to talk about today as more inflammation related to the innate immune system. I have put a T cell in here, just for balance, because we should always have balance in all talks. But I'm not going to talk about T cells. I'm going to talk about really the things that are produced by neutrophils and macrophages, and that interaction between neutrophils and macrophages and the production of things like DAMPs, calprotectin, cytokines, proteases, and the fact we have bacteria and mucus in the airway. And it's really the interaction of all of these things that characterize the non-resolving inflammation we see in CF. And essentially, non-resolving inflammation in the CF airway equates to damaged lung and lung function decline. So it's really something that we have to get a handle on if we're going to adequately treat our patients. So why is it important to be able to measure inflammation in patients? Well, it's important because patients, and this is pre-modulator era data, patients with higher levels of inflammation have worse clinical outcomes. So if we take patients in clinic who are otherwise clinically stable and we measure the levels of, in this case, calprotectin, a biomarker that we've worked on for a number of years in Edinburgh, if patients have a high calprotectin at baseline, even if they're clinically stable, they will exacerbate a lot sooner. So the level of inflammation you have measured systemically as a patient dictates how your outcome is going to be in a clinical sense over the next few months. So that kind of brings us to a major question, and that's if we restore CFTR function, can we resolve inflammation and stop lung damage and actually allow lungs to repair? And will this be due to a direct effect on CFTR function in immune cells, which we've seen displayed quite elegantly so far today, or will it be due to the effects of an improved airway environment with less stimuli for inflammation, which is also a possibility. And as we saw from the two talks related to microbiology today, that improvement or the changes in microbiology might be having direct impacts on inflammation. Also important to remember that we were able to modulate inflammation in some ways in CF before we had modulators. And this is data from the CF tracking study that was carried out in the UK over 10 years ago. And in this study, looking at patients who have extreme levels of inflammation during an exacerbation, if you treat these patients with antibiotics, their levels of inflammation improve after the antibiotic treatment. And we see this in a number of markers, including non-specific things like CRP, but also calprotectin and IL-6, which seem to be more specific to CF. I'd like to keep those biomarkers in mind as we're going through this talk, because they're probably going to come back in some of the later data, which I maybe wasn't expecting to start off with. So, what do we know already? What lessons did we have before ETI that we got from ivacaftor? You might recognize this person here from the audience, Dr. Heiser. That's me when I was a much younger person, and I didn't have vertical wrinkles on my face, I only had horizontal ones at that point. And that's Kelly, who was working with us in the lab. And in 2013, we were part of a group that went to Dublin to study the acute effects of starting ivacaftor in patients with G551D mutations and how this might affect their immune cells. And I was primarily looking at neutrophils, and Katie and Kelly were looking at monocytes. This was taken on a Friday night in a lab in Dublin at about 10 p.m. Most other people in Dublin were out in this beautiful city drinking Guinness, having a great time, whereas we were freestyling it with blood preps. Happy days, I think you'll agree, Dr. Heiser. Yes. Anyway, we've learned quite a lot from those studies and other studies people did at the time using ivacaftor as a tool to try and dissect some of the things that are going on with inflammation. So this is from Dr. Heiser's seminal paper in the Blue Journal, 2017. And what she's shown here in terms of inflammation is that there's an early change in inflammation measured by sputum elastase that happens in the first few days. And this actually continues over the next couple of years in these patients. So it's a sustained reduction in inflammation. There are also reductions in inflammation in other biomarkers in sputum early on, including S100A8 and A9, my favorite biomarker, calprotectin. The other quite fascinating thing they found in this study, though, was when they went out to 1 year and looked at patients in a radiological sense, that there was an improvement, particularly in mucus plugging in these patients, but also a suggestion that there was also an improvement in airway wall thickening. So perhaps that these patients who'd been on a modulator that was correcting CFTR at that point were actually having an effect on their structural lung disease. Of course, other groups have looked at ivacaftor and its effects on inflammation, and suffice to say, the findings were not consistent across all of the studies that have looked at ivacaftor. There are 2 studies here from the GOAL investigators in the United States, and in this study published in 2020, they didn't really show any significant changes in sputum markers, be they from induced sputum or spontaneously expectorated sputum. They did show that there was a trend towards some markers improving, but nothing significant. The same group have also now looked at the effects of ivacaftor in patients with gating mutations on serum measurements of inflammation, in this case the DAMPs HMGB1 and again our old friend calprotectin, showing that there's an early decrease in these measures of inflammation, and that's sustained over the following 6 months. We've also been able to use data from ivacaftor to tell us a little bit about the effects of CFTR modulation on immune cells. This again is some of Dr. Heiser's data looking at the membrane proteome of monocytes in patients who have had a short-term treatment with ivacaftor. And as you can see here, the membrane proteome changes, suggesting that there are changes in phenotype of these cells that are occurring with treatment. We were also able to look at neutrophil phenotype and how that might be affected by ivacaftor. And we've been interested in, for some time in Edinburgh, in the apoptotic phenotype in patients with CF, in that their neutrophils don't die as quickly as healthy control neutrophils. And we showed in that cohort of patients that ivacaftor treatment partially reversed that apoptosis defect. We did go back to Dublin to do a later study on R117H patients, and the important take-home message from that was that actually the R117H patients did not have the apoptotic phenotype that we saw in patients with more severe mutations, suggesting that it depends on how much CFTR activity you have, whether you will have this phenotype or not in the context of CF. So changes in neutrophil phenotype are happening. Other studies, of course, are available, and this was a really elegant study done at the time by Kirsten Paul, who also was from Dublin but from a different group, and she showed that ivacaftor treatment given to patients improved the functioning of this GTPase RAB27A, and that actually led to neutrophils from CF patients improving their bacterial killing. And other studies have looked at surface markers of phenotype of immune cells in both neutrophils and monocytes, showing that in some cases these will change. So really a body of evidence that if you modulate CFTR, you might actually see some changes in inflammation and in immune cell phenotyping. So the summary really from this introductory part of the talk was really just to give you an idea that we do know that CFTR modulation has an effect on things even before we get to data looking at ETI. So ivacaftor treatment in people with CF and gating mutations is associated with a decrease in systemic inflammation, but also that ivacaftor treatment changes monocyte and neutrophil phenotype. And that might signal direct effects of ivacaftor on CFTR function in these cells. So for the rest of the talk, I'm going to attempt to answer 3 questions, and I'll go through these in turn. So the first question is, what does elexacaftor, tezacaftor, ivacaftor, ETI do to inflammation in the first few months of treatment? The data I'm going to show you now is all unpublished. It was in the main as data that has come from the work of Nicola Robinson, who's sitting in the audience, who's an MRC training fellow in our lab at the moment. So the answer is that ETI does have a positive impact on systemic inflammation in the first 3 months of treatment. And we're looking at systemic inflammation because we could reliably get blood samples from these patients in the main who had stopped producing sputum. And importantly, we see a decrease in a general marker of inflammation, CRP. We also see a decrease in IL-6, and this is at 1 month, which is sustained to 3 months. suggesting that we're seeing something quite specific in CF because we saw no significant change in markers such as IL-8 and IL-10. We then, of course, looked at calprotectin, and what we found here was quite interesting. We saw a decrease in calprotectin in patients taking ETI, but if we divided these patients into those patients that were completely modulator naive, so patients who had never been on a previous modulator, they started off with a much higher level of calprotectin, as you might expect, and this decreased to a greater extent. Patients who had switched to ETI from previous TEZ-IVA therapy showed a trend, but not as significant a change, and that's probably because they'd already suppressed their level of systemic inflammation following a year or so of treatment with TEZ-IVA, which of course is not as efficacious a CFTR modulator, but was probably causing some effects in these patients to reduce inflammation. We also looked in these studies at immune cell phenotyping, and the honest answer from this is it wasn't particularly rewarding. If we look at baseline changes that we might expect, work from a PhD student in our lab worked up a panel of markers that were elevated in CF, suggestive of activated monocytes, and all we saw a significant change in ultimately was CD11b, suggesting that the cells were slightly less activated. We did see a trend towards CD14 expression decreasing, which might suggest that we have less classically activated monocytes circulating in these patients. But some signs of subtle changes in monocyte phenotype. And we also looked at neutrophil phenotype, again with whole blood flow cytometry. And in these patients, what we saw was actually an increase in CD16, which was significant, suggesting that perhaps the cells that were now circulating in patients after they'd started therapy were more mature than the original neutrophil populations that were circulating in our patients before therapy, and that would probably be supported by this trend, which is non-significant in CXCR2 expression. Another thing we did see was an almost complete reversal of the neutrophil apoptosis phenotype that was consistent with what we've previously seen in the G551D patients, but more significant. So these patients had elevated neutrophil or prolonged neutrophil survival, and we reversed that with ETI. This is in a small group of patients, but I think it shows the concept. So the conclusion from this very small first part is that we show a clear short-term improvement in systemic inflammatory biomarkers, suggesting that there's a degree of resolution of inflammation. But not all features of inflammation improve. So it's not as if every single inflammatory marker we measure changes. The changes seem to be quite specific, and they seem to center around IL-6 and calprotectin predominantly. So the second question is, what does ETI treatment do to cells involved in tissue repair? So if we ever want to make our patients truly better rather than halt their disease process, we want to have some effect on the repair machinery that is already present in their lung tissue, particularly airways. So airway repair occurs as a normal response to injury. It occurs in all of us consistently, and is contingent on basal cells that can migrate to the site of injury and differentiate into a wide range of epithelial cells that you'll see in the pseudostratified epithelium. And basal cells sit quite happily as a stem cell population in normal healthy airways. In CF, we know from previous work that has been done, you have an increase, basal cell hyperplasia, and these cells don't seem to work properly because you don't replace your airway with normal epithelial cells or the normal proportions. You have less ciliated cells and more secretory cells. And this is, again, data from Nicola, and what Nicola's done here is taken basal cells from the nose of CF patients and a healthy control patient. And healthy control patients make lovely, nice spherical organoids which then mature when grown in matrigel, but basal cells from CF patients don't do that. They form very small, abnormal organoids with abnormal morphology that don't really mature, suggesting there's a key issue with the basal cell in CF. So Nicola took this opportunity of our patients starting ETI to look in a small cohort of patients, I think it was 7 or 8 patients in this study, at nasal brushings before and after 3 months of treatment. And she's taken the nasal brushings and she's expanded the basal cells in culture. And you can see they're lovely looking basal cells, which are positive for cytokeratin 5. And in these patients, before and after, we see a significant difference in gene expression, with around 600 genes that were differentially expressed after 3 months of treatment in patients. And if we look at gene ontology analysis, we can see that several pathways are modulated, particularly positive regulation of cell proliferation, and perhaps most interestingly, positive regulation of the Notch signaling pathway. And that's important because Notch signaling is key to the way that basal cells will respond and proliferate into different sorts of epithelial cells to help the repair process in the pseudostratified epithelium. And here Nicola has just shown some validation data of these genes that were differentially expressed. And we think that this differential expression is now showing us a potential pro-repair signal in the CF basal cell after treatment. So, conclusion number 2 is that ETI treatment is associated with a pro-repair signature being induced in airway basal cells. And that might be a primary effect of the CFTR modulation in these cells themselves, or it might be something to do with the environment the cells came from. So, what does this all mean? And that brings us to question 3. What are the longer term effects of ETI on lung disease in CF? So, during the pandemic, we tried to do all of these studies, and it was not easy. And we planned to do a much larger single center study looking at CT outcomes in our patients. But our hospital at that point was refusing to do any research scans. So we managed to scan 10 modulator naive patients in our clinic, and we went back and scanned them a year later. And some of the changes we saw were really quite impressive. So this is a patient with very severe lung disease, as you can see, and they have airway wall thickening, they have mucus plugging, they have cystic disease. And after a year of therapy, some of the mucus plugging has disappeared, as previously shown by Pradeep in his talk today, but also a lot of the airway wall thickening has really disappeared. And some of these cystic areas have changed appearance. And we really weren't expecting to see CT changes as impressive as this after a treatment that simply turns CFTR function back on. And if we look at, see if this works or not. Yeah, this is just a fly through another patient with slightly milder disease. I wanted to get a sense of, there's lots of bronchiectasis here, but in the scan a year later, some of that bronchiectasis has actually disappeared. So there appears to be an actual remodeling process in some of our patients. So that damaging lung disease with related inflammation seems to be getting better, perhaps more than we could ever have expected. So in that cohort of patients, we decided, let's look and see if there's a signal. And this is a test group of 10 patients before and after 1 year of ETI. And we used an O-link proteomic analysis. So we looked at 184 proteins that were associated with inflammation and organ damage. And what we got was this Jackson-Pollock heat map, which shows no signal whatsoever as to some sort of key signature of proteins that was being changed by ETI therapy, which initially I was quite disappointed by and sent lots of emails weeping saying, oh, I can't believe we spent thousands of pounds on this analysis. But then we looked at the analysis a bit further. It turned out there were three proteins that were significantly different in these patients before and after 1 year. And these were IL-6, MMP9, and IL-20. And we're now validating these in a larger cohort. But again, IL-6 comes up as a top marker in these patients who have got significant improvements in their lung disease. And we have looked at that in our larger cohort, and it confirms that IL-6 is suppressed after a year on therapy with ETI, as is calprotectin. So going right back to the beginning of this talk, when I showed you data from patients with an extreme level of inflammation who had an exacerbation that we treated with antibiotics, in those patients, their levels of IL-6 and calprotectin improved, and we're seeing almost a mirror image of that with ETI therapy. So the conclusion of this last bit of the talk is that long-term treatment is associated with considerable improvements in lung disease on CT scan, and a sustained reduction is seen in biomarkers that we've previously known to respond to what I've called traditional CF treatments. So in terms of overall conclusions from this talk, as I've got a flashing red light, which means I've talked too long. ETI therapy is associated with improvements in inflammation. It also, we think, induces a pro-repair basal cell transcriptional signature. Longer-term therapy is characterized by improvements in structural lung disease on CT, and inflammation suppression persists over the long term. But this is only true for a select number of biomarkers, not all biomarkers. And it also potentially makes me think, are there treatment targets here for patients who can't take modulators, such as IL-6 and calprotectin? We do already have anti-IL-6 monoclonal antibody therapies. I'm not suggesting we immediately give those to our patients, but it's something we have to think about. Are there other drugs that might be repurposed to treat inflammation in patients that can't take modulators? So I was going to leave a question that we might bring to discussion, and that was ultimately what's causing the reduction in inflammation and our pro-repair signature and the clinical improvements in structural lung disease. Is it the effects on inflammation due to the lung environment improving? Because we've seen evidence of that today. Is it due to a specific CFTR effect on inflammatory cells? And we've also seen some evidence of that today. Or is it the classic multiple choice answer of C, all of the above? And it might just be that, we don't know yet. Just a quick mention of Nicola Robinson's talk on Saturday. She was hoping I wasn't going to do this, and she was hoping that everyone would have already gone to the social event by the time she's doing her talk at 4:30. But please go to her talk. It's important we encourage the young people who are doing CF research to get involved in these meetings and be able to present their fantastic work. And with that, I'll say thank you. The people who did all this work are these three here, Johnny, Gareth and Nicola. Johnny and Gareth have now departed because Johnny's decided to move to the continent, and Gareth decided he doesn't like lungs anymore and he's working on liver disease. So we're fairly depleted, but we are recruiting, so if anyone's interested, come and speak to us. And I want to thank my mentor, Moira White, and my great friends that I made in Dublin and all the people that have funded this work over the years. Okay, now we'll get on to the main part of the event.
Thank you, Robert. That was really great. So we'll bring back up our panelists. You can vote for who deserves a chair and who doesn't deserve a chair.
I'm going to go get some chairs.
We'll grab some. I can go up here, so that's fine. I can sit up here.
One more. One more chair.
There's four, right?
If you are.
I can moderate. I can do
Do you
four up
want to
here.
stand? Okay, yeah,
Yeah.
we're fine. Right, so we've got 4 chairs.
Okay, so we really appreciate the audience. We have a bunch of questions to go over, but I'm going to pose something for all the speakers. Because I think in most of the talks, we saw actually a lot of heterogeneity in responses, right? There was pockets of people that responded better, microbiomes that looked more dynamic than others, inflammatory signals different. Whereas, Pradeep, your data was actually a little bit more, homogeneous in terms of the responses, it seemed like. But just curious what everybody's thoughts on, how individuals are responding in terms of their pathogens, their inflammation. What are your thoughts?
So, well, I'll maybe start just by saying that the remarkable thing is that when we give these treatments to patients, most of them feel better. So regardless of what we show in terms of the assays, patients are feeling better. So it might be that you don't need a response in each individual facet. It might be that some people, their responses, their microbiology is better. Some people, it's their inflammation, but most people seem to feel better, and certainly that's borne out in all of the quality of life data that we've now seen and that's now being reported from the long-term clinical follow-up studies?
I would say that it's a really important question. We've been trying to look at predictors of response. Haven't done an in-depth analysis of patient characteristics that correlate with good microbiologic responses, but have looked at a number of factors. So in people who cleared Pseudomonas aeruginosa, was there some kind of micro predictor of that? We've looked at the presence of mucoid colonies, so categorizing people as having any mucoid colony or no mucoid colonies in their culture. The starting CFU counts that they had, and also looked at some other parameters like lung function and age. And so far, the only parameter that's showing a really strong signal, and that might change with time, is the starting CFUs. So if you have a CFU count in your sputum of Pseudomonas that's above, in general, 10 to the 6 CFUs or 10 to the 7 CFUs, your probability of converting to culture negative is very, very low. If you're at 10 to the 5 or so CFUs per mil, your probability of converting to culture negative is quite high. And that's interesting. It might argue, or it raises the possibility, I should say, that what really matters is, just bulk bacterial burden. A lot of us in microbiology are studying, pathogenesis functions, thinking about specific activities that bugs are doing to counter immune responses. And what really might matter is just, how many cells were there to start with. Those could be biomarkers or some other phenomenon, but just having a higher number of bugs could also produce phenotypes like slow growth or increased aggregate formation or increased stress response induction by the bacteria because of the high density. So that's at least one factor that we've identified so far as showing an association with response.
So I guess I have a little bit of a stream of consciousness with this question because there's so many things playing a role, and I think when you look at this topic, it's so important to look at people longitudinally and what they do, and not only is there this immediate change, right, but then there are changes in people who have an immediate improvement who continue to have stability, and then they will then lose stability. And I think that's related to pathogen, and I think that's related to immune response, and of course those are very tightly connected. I am thinking in particular of a person who I showed who went from 6 admissions the year prior, she was our star, 6 admissions the year prior to starting Trikafta to 0 in the year following Trikafta. She had incredible changes on her single cell, her Cystic data, and then the year following that, she had 4 admissions. And so then, what happened there? Was it her bug? Was it her immune response changing? And so one of the things that we have tried to do in coordination with our human data is to do targeted amplicon sequencing of the microbiologic data as well. And that's going to be presented tomorrow in a session in which we look at different things that are happening in terms of virulence and antimicrobial resistance in patients over the time before and after Trikafta. And one thing that you can see in that particular patient is that there was very little change in her Pseudomonas the year prior and the year after in terms of AMR genes, in terms of virulence. She lost her staph. So perhaps that was its initial effect of she lost staph, but then she maintained that Pseudomonas kind of in its same exact state where she kind of had a bump, and then she then got sick again more frequently, versus a patient who actually cut her admissions by a third. And that patient, when we evaluate by targeted sequencing, basically had lost her whole PSL operon. and that person has maintained their improvement. And so I think you have to think of so many things when you're thinking about this. You think, the pathogens themselves, who stays, who goes, how do the pathogens change, and then, what's happening to the immune response in general.
Could I ask you a question about that? Mm-hmm. One of the things that I find quite fascinating is we started these drugs predominantly during a global pandemic when our patients were isolating, et cetera. Do you think that's had an impact on your T-cell biology as well? Because ordinarily these patients would be getting exposed to viruses on a day-to-day basis. Because I do wonder if the reason we saw such an amazing change in inflammation and CT scans in our patients where we essentially had those patients locked in a room for a year, gave them a highly effective treatment, and then didn't expose them to any environmental stimuli.
Yeah, yes, absolutely. I think that plays a huge role, and so part of the way that we've addressed that is to have data at 1 year after starting Trikafta, which pretty much would have gotten all of 2020, and then a year following that, and then comparing each year, because there's a big change. Some of it's from respiratory viruses in this most recent year.
So we've talked a little bit about some of the heterogeneity and the responders and non-responders. We had several questions from the audience, related to that. I think the COVID pandemic was something that came up certainly with the bacterial pathogens in terms of, what's there and what's not there. But we have several questions also kind of related to the anti-inflammatory, the resolving signature. I know, Robert, you talked a little bit about this, but what about, have people looked further into details in relation to, the metabolites that are produced, other, kind of pro-resolving cytokines? Do you guys have any thoughts on how those are changing as this landscape is shifting?
I mean, mostly just what we showed, but excuse me, what I tend to think about in this context is one of the strongest phenomena in biology is positive feedback loops, right? So global warming lowers the glaciers reflect more sun and makes the earth hotter. So if everything is sort of working collectively to reverse what we think of CF airway pathophysiology, that might explain the response. My understanding from speaking to physicians is that it was even a more positive impact than might have been anticipated. And it might be just that the whole system has crashed, right? Where you have these long evolved pathogens that are very persistent, have been there for a long time, and all of a sudden everything that they evolved to be exposed to is being reversed, and that might be explaining. Just think about the metabolites. The amino acids was the major signature we saw. We didn't see changes in lipids, type of phospholipids, so it's not just a total reduction of everything. There's kind of a chemical change in the sputum that we saw. Now, the biggest telling point is the chemical change in sputum is that it's gone, right? We can't even get it anymore, so.
That's an interesting point about the positive feedback, and I was thinking the same, that we might see a self-amplifying effect. You could imagine that you reduce pathogen burden markedly, that you improve airway physiology, you improve host defense, and what I expected to see in the pathogen tracings was a continued improvement over time. And surprisingly, we didn't see that. What we saw was this very marked, very consistent, approximate 100-fold reduction in almost all the traditional pathogens occurring within a 1-month time frame, and then marked stability after that. And I like the way you thought about it, Robert, and that's why I was thinking we would see too, that there would these improvements would feed back on each other and produce continual improvement, and at least in pathogen burden we don't see that.
I think just to mention something about the potential pro-resolving cytokines, and that small sort of proteomic study we do, we did look at that, and it was just quite remarkable that we didn't see changes in more of those cytokines. And whether that's because the inflammation that we were measuring was so extreme, coming back to a more level of normality, that we're unable to see that, or I don't really understand it to a certain extent. It does make me think, however, that a lot of the changes in that systemic measurement of inflammation are probably due to the reduction in bacterial burden, as shown by Pradeep, because the results do mimic what used to happen in our patients when we gave them antibiotics for 2 weeks and significantly reduced bacterial burden before things rebounded. I've got no doubt there are also CFTR effects on immune cells. So Johnny, my student, couldn't show all of his data today. He's just submitted a paper where he used cage sequencing of CF and non-CF derived macrophages and showed that there was a major shift in type 1 interferon signaling in the CF macrophages, particularly after LPS, and that can be corrected with ETI in vitro, and we can also correct it by correcting CFTR with CRISPR. So there are definitely things that are going on at an immune cell level, but it might be that a lot of that is being drowned out by just the sheer response in terms of clearing bacteria, which is really impressive to think that you've reduced that burden so extremely in that first month. So yeah, that was just an additional point.
Yes, I mean, I think it's really a fascinating area, and we still have a lot to think about. So currently the most popular question is a clinical one, and I think it's based on some of these shifts we've seen with, pathogens down, mucus down, inflammation down, neutrophils less recruited. So what do you guys think in terms of, in the audience, feel free to chime in as well. I know we have some good pediatric and adult representation here. Is Pulmozyme or medicines like Pulmozyme going to be as useful for folks on highly effective modulators? Do we really need to start thinking again about what types of suppressive therapies we're using?
Do you want the anecdote? I mean, we can give you clinical anecdote, which is that undoubtedly the patients that have stopped taking their DNAs, I think their lung function is not quite as good a year down the line as the patients who have continued to take it. And obviously that's being assessed at the moment in the CF STORM study, which is a UK-wide study looking at mucolytic removal. I've always been a great believer that DNase was more than a mucolytic. Good evidence previously that it's an anti-inflammatory or it has anti-inflammatory activities. It certainly would fit with the fact we know there are lots, or there used to be before when we could measure them, lots of nets in the CF airway, and DNase would be treating those. So my feeling is that it's probably still has a place, and it's probably going to have a place for a lot of patients. And again, we'll find out more as time goes on, particularly as we've come out of the pandemic now, that we had a lot of patients who just were stopping everything. The other thing to remember about a lot of our patients as well is they weren't actually taking it in the first place. They just told us they were taking it. And so that also confuses matters. So we had a lot of our patients had the biggest changes in lung function with these drugs. When we retrospectively thought about it, they were our patients who they weren't taking their standard therapies because they were trying to get on with life and not think about their CF. So that comes into all of this as well.
I don't have any data to respond to the question on. I would maybe even take a one step backward or a broader question, and in addition to do anti-infective therapies or anti-inflammatory therapies need to be continued, I think we need to ask the question, does infection matter anymore? So if you have improved inflammation and perhaps have reduced the pro-inflammatory effects of CFTR mutation itself, you've cut back pathogen burden by 100-fold. Do people who are persistently infected really having worse clinical outcomes than those who aren't? To me, that's a key question that could drive future research.
Yes, I agree with you very much. And it's interesting because, like you just mentioned, this anecdotal kind of patient experience thing is there will be patients who have basically don't grow their pathogen by traditional culture anymore, and there will be patients who grew 10 to the 7 CFUs per mil of Pseudomonas before and 10 to the 7th or 10 to the 8th after, and clinically they feel the same. and they may not, produce very much sputum. They may feel fine. I don't know, the answer to that. Well, I won't even try to answer that, whether, should the patients who still have clinically detected bacteria, should we approach them differently, especially if their disease is now slowed down to the point that you don't really see a phenotypical difference in terms of disease progression. I guess I'll finally answer that question with another question, and that is this whole issue of inhaled antibiotics. We're studying, obviously Simplify, where we're taking away pulmozyme, we're taking away hypertonic, but what about inhaled antibiotics, which many patients have stopped, and so I think that's why this whole longitudinal understanding of what happens in infections, what's their natural evolution over time is incredibly important in this time, so we can advise them properly in the clinic setting, which I struggle with myself all the time.
Just what you mentioned, I was thinking about the chronic therapies of antibiotics. I mean, they don't work very well, right? Like, we have 10 to the 9 Pseudomonas in the lungs of CF patients for like 30 years, taking kilograms and kilograms of antibiotics. And you just take a new drug that changes the entire system. And then, as Pradeep showed, Pseudomonas just drops massively, what antibiotics could never achieve.
Yeah, I mean, I think it's an interesting thought to think about. Do we need to revisit things that have failed before or maybe didn't work as well? In this landscape, we might have a better chance of utilizing some of those therapies. So, all right, Mimi, I'll keep you there. I think your data set was really interesting because we don't have much on T cells and adaptive immune function CF to begin with. But Rabin was curious, so you show the presence of TRMs in the blood, which has not really been shown by a lot of other prior Cystic studies and non-CF. So there's suggestion that they're not really very easily released from the vascular beds. Do you think there's something different that's leading to the release that you saw? Have you guys looked into other, data regarding the TRMs? So the, in terms of like the TRMs, that I didn't show all the percentages, but that literally was maybe 1% and 2% of our total T cell genes. And so our immunology expert, which is Lee Reinhardt, he kind of helped me understand all this. And so apparently you can have very, very, very small amounts of TRMs in the circulation. Obviously, I'm not going to be able to give you any kind of sense of what's actually going on at the level of the tissue under the mucosa and what those, T cells are doing, or in the adjacent lymph nodes or in the ball tissue. But it's this issue of creating a model system that's evaluable easily to understand what's happening with this kind of leg of immunology. And I think for CD8 cells, it's going to be trickier. What they're doing locally in the tissue is probably different from what that cell's reflecting in the peripheral blood versus a CD4 cell where, you may see a little bit more homology and be able to kind of make predictions. So it differs across the different CD8 cells. But yes, I would say, hard to make any major conclusions, and they were very, very, very lightly represented in our population. Okay, so I know we have a lot of questions that we didn't get to, so I'll encourage everyone out there to connect with the speakers afterwards. I will make sure we show everybody the stuff that was in the chat as well. So I think it's been a really great discussion. I know everybody's anxious to keep going on this meeting, but really appreciate everyone's thoughts, and I think there's a ton swirling in our heads right now about what to do moving forward, and we really didn't even touch, what do we do now that we don't have sputum? How do we measure all this? So I think that's going to be in one of the other sessions at the conference. So thanks again, everyone.
Thank you.
Thank you.